Hello students, this is Dr. Tiki Arashiro. In this podcast, I'm going to talk about mixed acid fermentation and butyric acid fermentation. So let's start with mixed acid fermentation. Mixed acid fermentation is a type of fermentation process that results in the formation of variable mixture of acids such as lactate, acetate, succinate, formate, as well as alcohol and gases such as hydrogen gas and carbon dioxide. The formation of these end products depends on the presence of certain key enzymes in the bacterium. The proportion in which they are formed also varies between different bacterial species. Mixed acid fermentation is carried out by enteric bacteria. These are gram-negative bacteria that belong to the family Enterobacteriaceae. They are facultative anaerobes. At least 40 genera have been identified in this family, including Salmonella, Proteus, Serratia, Enterobacter, Citrobacter, Klebsiella, and Escherichia. These mixed fermenting microbes start off with glycolysis to produce pyruvate. From pyruvate, several acids can be generated. So let's talk about the growth metabolism of enteric bacteria. Enteric bacteria have a very interesting growth and metabolism. First, the composition of the electron carriers in the electron transport system of these enteric bacteria varies depending on the electron acceptor available. Because enteric bacteria are facultative anaerobes, the terminal electron carriers in the electron transport system varies depending on the presence or absence of oxygen. Terminal oxidases such as cytochrome O are expressed when oxygen is present, while terminal reductases such as nitrate reductase are expressed when oxygen is absent. Secondly, the type of citric acid cycle they use varies depending on the availability of oxygen. When oxygen is available, Enterobacters utilize the oxidative citric acid cycle. On the other hand, when oxygen is depleted, they do not express the alpha-glutarate dehydrogenase enzyme and use the reductive or incomplete citric acid cycle instead. Thirdly, Enterobacters are not only capable of switching from aerobic to anaerobic respiration, they are also capable of switching from anaerobic respiration to fermentation when oxygen is absent. So when oxygen is present, they use aerobic respiration, and when oxygen is depleted, they switch to anaerobic respiration if there are suitable alternative electron acceptors such as nitrate, fumarate, or trimethylene N-oxide that they are able to utilize. In the absence of these electron acceptors, they will switch to fermentation and ferment glucose to a mixture of acetate, formate, lactate, and succinate acids plus ethanol. Some will carry out butanediol fermentation. So what's the difference between mixed acid fermentation and butanediol fermentation? Well, they vary in terms of the main products they produce. In mixed acid fermentation, acids are the main products. 4 acids are formed in significant amount, and they are formate, lactate, succinate, and acetate. Ethanol, carbon dioxide, and hydrogen gas are also formed, but they are produced in small amount. Because acids are the main products, the fermentation process lowers the pH of the medium to 4. In butanediol fermentation, alcohols such as butanediol and ethanol, as well as carbon dioxide and hydrogen, are the main products. Acids such as lactate and formate are produced in small amount. Note that acetate and succinate are not produced in butanediol fermentation. Because small amount of acids are produced, the pH in the fermentation product is 6. So the products of mixed acid fermentation are succinate, lactate, acetate, ethanol, formate, carbon dioxide, and hydrogen gas. Succinate is made from carboxylation of phosphoenol pyruvate to oxaloacetate, followed by 2 reductions. Lactate, acetate, ethanol, and formate are made from pyruvate. The formation of lactate from pyruvate is simply a reduction reaction. Pyruvate is also decarboxylated to acetyl-CoA and formate by means of the pyruvate-formate lyase. The acetyl-CoA can be reduced to ethanol or converted to acetate and ATP via acetyl phosphate. The formate can be oxidized to carbon dioxide and hydrogen gas by the enzyme system formate hydrogen lyase. This system actually consists of two enzymes, the formate dehydrogenase and an associated hydrogenase. The formate dehydrogenase oxidizes the formate to carbon dioxide and reduces the hydrogenase, which transfers the electrons to two protons to form hydrogen gas. The butanediol fermentation, on the other hand, is characterized by the production of 2,3-butanediol and acetone. Glucose is oxidized via the glycolytic pathway to pyruvate. There are three fates for the pyruvate. Some of it is reduced to lactate, some is converted to acetyl-CoA and formate, and some is used for the synthesis of 2,3-butanediol. The formate is converted to carbon dioxide and hydrogen gas, and the acetyl-CoA is reduced to ethanol. The first free intermediate in the butanediol pathway is alpha-acetolactate, formed by the enzyme alpha-acetolactate synthase, which decarboxylates pyruvate to enzyme-bound active acetaldehyde. This is a reaction that depends upon thiamine pyrophosphate. The active acetaldehyde is transferred by the alpha-acetolactate synthase to pyruvate to form alpha-acetolactate. The alpha-acetolactate and alpha-ketocarboxylic acid is decarboxylated to acetone. The acetone is reduced by NADH to 2,3-butanediol. The production of butanediol is favored under slightly acidic conditions, and it is a way for the bacteria to limit the decrease in external pH caused by the synthesis of organic acids from pyruvate. Now I want to go over IMVIC test. IMVIC test is used to differentiate among enteric bacteria. This is a series of 4 different tests: the indole test, the methyl test, the Voges-Proskauer test, and the citrate test. The methyl red test determines whether the organism produces mixed acids or is capable of mixed acid fermentation, and the Voges-Proskauer test determines whether the organism produces butanediol or is capable of butanediol fermentation. So Escherichia, Salmonella, and Shigella are mixed acid fermenters and test positive for methyl red test, while Serratia, Erwinia, and Enterobacter are butanediol fermenters and test positive for Voges-Proskauer test. So the methyl red test determines whether the organism produces mixed acids or is capable of mixed acid fermentation. In this assay, methyl red is a pH indicator. Cells are grown in MRVP medium and then incubated. After incubation, 3 drops of methyl red indicator solution are added to the tube without shaking or mixing. The development of a stable red color in the surface of the medium indicates sufficient acid production to lower the pH to 4.4 and constitutes a positive test. The mixed acid pathway gives 4 mole of acidic products, mainly lactic acid and acetic acid, 1 mole of neutral fermentation product in the form of ethanol, 1 mole of carbon dioxide, and 1 mole of hydrogen gas per mole of glucose fermented. These large amounts of acid result in significant decrease in the pH of the medium below 4.4. This is visualized by the pH indicator methyl red, which is yellow above pH 5.1 and red at pH 4.4. The appearance of a color on the surface of the medium indicates a pH of 4.4 or below. The pH at which methyl red detects acid is considerably lower than the pH for other indicators used in bacteriologic culture media. Thus, to produce a color change, the test organism must produce large quantities of acid from the carbohydrate substrate being used. So a methyl red negative is when the culture medium remains yellow after addition of methyl red, and this occurs when less acid is produced. In this example here, E. coli is positive for methyl red test, indicating that it is a mixed acid fermenter, while Enterobacter aerogens is negative for methyl red test, indicating that it is not a mixed acid fermenter. The Voges-Proskauer test determines whether the organism produces butanediol or is capable of butanediol fermentation. In this test, cells are grown in MRVP medium and then incubated. After incubation, Barrett's reagents are added. Barrett's A is a reagent consisting of alpha-naphthol dissolved in ethanol, and Barrett's B reagent consists of 40% potassium hydroxide. A positive test is represented by the development of a red color 15 minutes or more after the addition of the reagents, indicating the presence of diacetyl as the oxidation product of acetoin. Organisms such as Enterobacter aerogenes produce acetoin as the chief end product of glucose metabolism and form smaller quantities of mixed acids. In the presence of atmospheric oxygen and 40% potassium hydroxide, acetoin is converted to diacetyl, and alpha-naphthol serves as a catalyst to bring out a red or pink complex. So let's talk about butyrate fermentation. Butyrate fermentation is a fermentation process in which butyric acid is the fermentation product. Butyrate fermentations are carried out by butyric acid clostridia, including Clostridium butyricum, Clostridium cluiveri, and Clostridium pasteurianum. Clostridia are a heterogeneous group of anaerobic spore-forming bacteria. They can be isolated from anaerobic muds, sewage, feces, and other anaerobic environments. Some are saccharolytic and can degrade carbohydrates. Some are proteolytic and can degrade proteins. The proteolytic Clostridia are important in anaerobic decomposition of proteins. Now, most butyric acid Clostridia ferment carbohydrates to butyric acids. The fermentation products also include hydrogen gas and carbon dioxide. Other Clostridia are specialized and will ferment a few substrates like ethanol, acetate, certain purines, or certain amino acids. Butyric acid is used in the manufacture of cellulose acetate butyrate plastics. It has also been tried for its potential use in the treatment of leukemia and sickle cell anemia. So here is the butyrate fermentation pathway. As you can see here, the pathway begins with glycolysis. The butyric acid Clostridia ferment glucose to butyric acid. The fermentation products include hydrogen gas, carbon dioxide, and a small amount of acetate is sometimes produced from pyruvate by some species. So the butyric acid Clostridia first oxidize the glucose to 2 moles of pyruvate via glycolysis. This process produces 2 molecules of NADH plus 2 molecules of ATP. The pyruvate is then decarboxylated to acetyl-CoA, carbon dioxide, and hydrogen gas by means of pyruvate ferredoxin oxidoreductase and hydrogenase. The acetyl-CoA is condensed to form acetoacetyl-CoA, which is reduced to beta-hydroxybutyryl-CoA by means of one of the 2 NADH generated in glycolysis. The beta-hydroxybutyryl-CoA is reduced to butyryl-CoA by using the second NADH. The CoAsh is displaced by inorganic phosphate, and the butyryl phosphate donates the phosphoryl group to ADP to form ATP and butyrate. So the pathway produces 1 mole of butyric acid, 2 moles of carbon dioxide, and 2 moles of hydrogen gas per mole of glucose. It also regenerates 2 NADHs. Some Clostridia initially make butyrate during fermentation, and when the butyrate accumulates and the pH drops owing to the butyric acid produced, the fermentation switches to butanol acetone fermentation. So butanol acetone fermentation is a type of fermentation in which butyrate is initially produced and then followed by a mixture of butanol and acetone as fermentation products. An example of Clostridia capable of butanol acetone fermentation is Clostridium acetobutylicum. Clostridium acetobutylicum is a bacterium that ferments sugar to a mixture of organic solvents like acetone and butanol. The organism uses the glycolytic pathway to convert glucose to pyruvates. Then it ferments pyruvate to butyric acid. When the butyric acid accumulates and the pH drops to about 4.4 due to the butyric acid accumulation, the fermentation switches from butyric acid fermentation to butanol acetone fermentation. The butyrate produced in the initial butyric acid fermentation is actually taken up by the cell and then converted to butanol and acetone via butanol acetone fermentation. The accumulation of butyric acid in the culture media of low pH can be toxic because of the undissociated form of the acid is lipophilic and can enter the cell. Butanol and acetone production by Clostridia was one time the second largest industrial fermentation process after ethanol. The solvents are now synthesized chemically. As an example of the butyrate fermentation and the shift to the butanol acetone fermentation, we will consider the fermentation of carbohydrates carried out by Clostridium acetobutylicum. The fermentation of carbohydrates by Clostridium acetobutylicum has two phases, the acetogenic phase and the solventogenic phase. The exponential growth is what is called the acetogenic phase. And in this phase, the bacteria produce butyrate, acetate, hydrogen gas, and carbon dioxide. Now, when the culture enters the stationary phase, the acids are taken up by the cells, concomitant with the fermentation of the carbohydrate, and are converted to butanol, acetone, ethanol, and carbon dioxide. And this phase is called the solventogenic phase. This figure shows the butyrate and butanol acetone fermentation in Clostridium acetobutylicum. As you can see here, glucose is oxidized to pyruvate via the glycolytic pathway or the Embden-Meyerhof-Parnas pathway. Then pyruvate is oxidized to acetyl-CoA. Pentoses are also fermented, and these are converted to fructose 6-phosphate and phosphoglyceraldehyde via the pentose phosphate pathway. In the acetogenic phase, the acetyl-CoA is converted to acetate and butyrate. So some of the acetyl-CoA is converted to butyrate and ATP. Some acetyl-CoA is also converted to acetate via acetyl phosphate in a reaction that yields an additional ATP. Now, in the solventogenic phase, the butyrate and acetate generated from the acetogenic phase are taken up by the cells and reduced to butanol and ethanol. In this process, the acids butyrate and acetate are converted to their CoA derivatives acetyl-CoA and butyryl-CoA by accepting a CoA from acetoacetyl-CoA. And this converts acetyl-CoA to acetoacetate. Now, the acetoacetate that is formed is decarboxylated to acetone and carbon dioxide. The acetyl-CoA produced from acetate is reduced to acetaldehyde and then to ethanol, and the butyryl-CoA produced from butyrate is reduced to butanol. The acetogenic phase produces 1 mole of butyrate, 1 mole of acetate, 1 mole of lactate, 2 moles of carbon dioxide, and 2 moles of hydrogen gas. The solventogenic phase produces 1 mole of acetone, 1 mole of ethanol, 1 mole of butanol, and 1 mole of carbon dioxide. The pathway as a whole generates 4 moles of NAD and 1 mole of NADP. The microbial production of acetone and butanol was one of the first large-scale industrial fermentation processes of global importance. Acetone has been used for the production of cordite, which is a smokeless munitions powder used during the First World War. Butanol, on the other hand, has been used as a biofuel. It is actually a more efficient biofuel than ethanol. Now, to summarize butyrate and acetone-butanol ethanol fermentation, the Clostridia cells break up glucose to pyruvic acids via glycolysis. Now, the pyruvic acids generated then go through butyrate and then acetone-butanol ethanol fermentation. During the exponential phase of growth, butyric acid, lactate, and acetate are produced and released in the culture media. When these acids accumulate, the pH drops to about 4.4. Then butanol acetone fermentation begins. In this process, the cells take up the acids and convert them to acetone, butanol, and ethanol, and then secrete them into the media. You are probably wondering why the organism switches to solvent fermentation when the pH drops to 4.4. It switches to solvent fermentation because the accumulation of butyric acid in the culture media of low pH can be toxic because of the undissociated form of the acid is lipophilic and can enter the cell. So to prevent toxicity, the organism used the acetone-butanol ethanol fermentation to bring the pH back close to neutral. So what are the consequences of excessive accumulation of butyric acid in the cell? Excessive accumulation of butyric acid causes toxicity due to the fact that the undissociated form of the acid is lipophilic, which can enter the cell and cause cell death. Butyric acid accumulation decreases cytoplasmic pH. Recall that cytoplasmic pH is about 7.8. So when butyric acid accumulates in the culture media, the cytoplasmic pH will shift to acidic. This shift in pH will affect the functioning of the enzymes in the cell. So the acetone-butanol ethanol fermentation process neutralizes the pH by converting the acids to solvent, thus increasing the cytoplasmic pH close to neutral. Butyric acid accumulation also results in acid crash or overproduction of undissociated acids. Acid crash leads to the cessation of glucose uptake and fermentation failure. That's the end of the third podcast on fermentation. Stay tuned for the fourth podcast.